So last time, I would have gotten to talking about antibodies. I talked about IgMs and IgGs and that stuff, right? The table of the 5 different types. Okay. That, if I remember correctly, that's sort of the last thing I covered. Okay. Apparently Danielle's the only one that was here. So I just want to cover one last thing and then just say a few closing remarks about the material. The one last thing that I want to say, that I want to cover is actually not in the book, which is really sort of unfortunate. It is kind of important to understand what happens after an antibody binds to an antigen in general. So hold on. All right, so apparently the computer decides to be a little bit slow tonight too, which just adds to the frustration of getting a late start. I don't know why it's so slow in loading a freaking blank page, but okay, come on. So while we're waiting for this to get around doing its thing, what I want to talk about here is when an antibody binds to its antigen, which may or may not be a pathogen, whatever it binds to. There's a few different things that can happen, and different sources I've used, textbooks I've used, have presented it in slightly different ways, but they all usually get the same thing. And the way that I've become comfortable with presenting this, too big, is based on a mnemonic Plano. Now, actually, when I first started doing this, the mnemonic I used, which I got from somewhere else, was just plan. But over the years of presenting this, I decided that I needed this last part of it, which is an O. So each of these letters stands for a particular thing. P, L, A, N, O. Whoops, O. P stands for precipitation. Okay. Now, when you hear the word precipitation, what comes to mind? Yeah, rain. Okay. What precipitation means is something falls out of, so to speak, solution. Now, with rain, what that kind of means, and this is a little stretch, because in this sense, I'm talking about it in a very chemistry-based definition, which is not exactly what precipitation in terms of rain means, but we can kind of make it fit. Water vapor is in the clouds. And when water molecules start to aggregate more and more, the water vapor becomes liquid water. And at that point, it's too heavy to stay in the clouds, and so it falls from the cloud. That's rain, that's precipitation. In a chemical sense, precipitation usually describes when you have a chemical in solution and something changes that chemical and it no longer stays dissolved. I can't remember now, but there's a rather classic example of this, which I'm kind of making this up off the top of my head. I don't remember the exact chemicals, but I think something like this. If you put Sodium chloride and there's some silver salt, and I don't think it's silver nitrate, but some silver compound. If you mix that with sodium chloride, the sodium and the silver sort of swap places. So you get, well, if I'm saying this right, which I think I'm wrong, you'd get, say, silver chloride and sodium nitrate. And I'm pretty sure sodium nitrate is actually explosive, so I must be wrong. But let's pretend I'm telling the truth. So the silver compound now that's made by mixing this other silver compound with salt does not dissolve in water. So in the beaker, the test tube where you're doing this reaction, you add salt and it dissolves in. You add this the silver compound, it dissolves in. But then when they mix, the new compounds, one of them no longer dissolves in water, and so it falls out of solution. You see a little white powder kind of thing at the bottom of the test tube or the beaker or whatever. That's called a precipitate. When we're talking about antibodies and antigens, precipitation is when a small antigen falls out of solution. When I say small antigen, what I mean here is like a protein. Okay, so it could be my sort of go-to protein antigen is snake venom toxin. So if that's in your system and you've taken the ampule of anti-venom serum, then the antibodies that you've introduced to your system will bind to the snake venom toxin, and that small protein that dissolved in your blood will no longer dissolve because it's now part of a bigger complex that includes the antibodies, and so it precipitates out, which removes it from the plasma as a solution and makes it easier for you to deal with that. L stands for lysis. Lysis is what we call destroying a cell. A more complete word is cytolysis, and cyto means cell. So cytolysis is breaking a cell. This is what complement does. Okay, so we looked at complement before, and when the membrane attack complex is inserted into the bacterial membrane, the cell is destroyed. This is the classical, I should put it, classical complement pathway. Lysis is also the result of the alternate complement pathway, but the classical complement pathway is the one that's initiated by antibodies. The alternate one is not. So this is specifically the complement, classical complement pathway. And then A stands for agglutination. And this is sort of the same thing as precipitation, but for large antigens, meaning cellular. Running out of space there, so I'm just abbreviating the word solution. This is often thought of as clumping also, and this is what we talked about with blood typing. When you find type A blood with A antibodies in the testing of blood type, agglutination is what happens. Red blood cells are all attached to antibodies, and you get this big cluster of cells, clump of cells that you can see in the reaction in the test. If this is happening in your body, if you have some sort of cellular antigen that's gotten into your body, which might be an incorrect blood transfusion, or it could be a cellular pathogen. Probably more like an amoeba or something like that rather than bacteria. Just like small antigens are precipitated out of the blood, large antigens agglutinate, and therefore they're no longer going to just travel through the blood easily. Okay. N equals neutralization, which is to make the antigen ineffective. A good example of this is if you get viral particles into your system, if they're suddenly covered in antibodies, then the virus particle itself can't interact with the host cell that it's trying to infect. Sort of like if you were wrapped in bubble wrap, it'd make it very hard for you to interact with the world around you. Okay. An outer covering of stuff removes you from the surrounding environment, and you can't do what you would normally do. Okay. I don't know where the bubble wrap idea came from just now, but the idea is just the antigen's covered. It could be a small antigen like a protein, and if it's covered in antibodies, it won't be able to do whatever it's supposed to do either. There are some bacteria that are motile, and they have flagella, and if antibodies bind to the flagellum mechanism, not necessarily all over the bacterium, but just to that, and it can no longer move, that's neutralized too, because it can't move in your body. Okay. So there's different ways that we can describe neutralization. It's just the antibodies interfere with the antigen's ability to do whatever it's supposed to do. And then finally, O, which I actually added to this list, stands for opsonization. And that means, oh shoot, what's the name? Enhancing phagocytosis. Now, in the innate immune response part of the book, I actually glossed over where it mentions opsonization because it wasn't really what I was trying to cover at that time. So back when I was talking about the interferon signal that virally infected cells release, that was in the section that was called early activation proteins or something like that. In that paragraph, it also mentions opsonization. Opsonization is just a very general term, anything that enhances phagocytosis. So if something in your system, which might be part of the complement system or some other chemical, helps to mark the antigen as something that needs to be phagocytosed, that's opsonization. Antibodies happen to do that. When they attach to the surface of an antigen, they have a little marker on them that interacts with phagocytes and makes it easier for the phagocyte to do its job. So that's opsonization. Now, like I said, I added this on to the list because it's something I want to mention by itself. But part of the reason I did that is just talking about PLAN, all of these things end with phagocytosis. So when small antigens fall out of solution, where they go is macrophages come along and gobble them up. When bacteria are lysed by complement, then phagocytes come along and gobble up the debris. When large cellular antigens are agglutinated, then phagocytes come along and gobble them up and get rid of them. When antigens are neutralized, then they're going to be cleared away by phagocytes coming along to gobble them up. So all four of the other things end in phagocytosis because the antibodies that are bound to things enhance that phagocytosis. But it's possible that opsonization can be the only thing that happens, okay? If antibodies start binding to something, but it never gets to the point of activating the classical complement pathway, or it never gets around to causing the small antigen or the large antigen fall out of solution, it can still be phagocytosed. So it goes right to the opsonization part. So that can happen by itself. Okay. But these are all the different things that can happen once an antibody binds to the antigen. Okay. So, like I said, that's not exactly laid out so clearly in this section here. They concentrate more on what antibodies are, their structure, the different types, which is all fine. And then they go into how B cells are activated and their clonal selection, meaning the ones that are activated make copies of themselves, which become plasma cells. And then they go into the primary and secondary response stuff. And then on to active and passive immunity, which I talked about a while back. The day that we looked at or listened to the podcast, I talked about active and passive immunity a little bit. But so the book never really gets into these series of things that can happen with the antibody antigen complex. But it's important to know what happens. It's not like the antibodies are there and the world is saved and everyone's happy. There's more to what happens when they show up, and that's what this is about. Now, when I was showing you this picture, did I talk about primary and secondary responses? I did? Okay. Just want to be sure. I wasn't sure if I did that in all my classes. But at the top of this, it says primary and secondary response. And down below, here's a graph that presents the idea. Just the y-axis is the concentration of antibody in the system, and then time along the x-axis. And so from when you're initially exposed to something like chickenpox virus, you develop a response to that, which will help fight off that initial infection. It's not a very robust response, and you're going to experience the symptoms of chickenpox and that sort of thing until the immune system finally fights it off. But the memory cells that are generated in that initial activation of the B cells, or the T cells have memory cells too, are going to be available. So the next time you're exposed to the same antigen, you have an immunological memory of it, and you can immediately start producing antibodies, and the level of antibodies reaches the equivalent of the primary response very quickly, and it continues making more to a much higher level. Now, it's a little unfortunate. A lot of textbooks will kind of oversimplify this figure. I think it's important to realize the scale that's on the y-axis here is logarithmic, okay? Which means it's an exponential scale. It's not linear. If we had tick marks here, it wouldn't just be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 up the scale. It would be 1, 10, 100, 1,000, 10,000, et cetera, up the scale. I mention that because the difference between the primary and the secondary response that's here is actually huge compared to what the picture suggests. If we're talking about some arbitrary number of or concentration of antibodies, if the primary immune response is 1 arbitrary unit of concentration, the secondary response is going to be something much more along the lines of thousands or 10,000s of units.
Dun, dun, dun.
Okay. So, just what this figure is showing is actually a huge response that happens for the secondary response. Now, primary response is the first time you're exposed to something. The secondary response is really all subsequent exposures. So if this is chickenpox, the first time you get chickenpox, you get sick. But then if you're ever exposed to chickenpox again, whether it's one more time in your life or 20 more times in your life, you're always going to have this secondary type response, and you're going to not have any symptomology. You're going to fight off the infection before it's ever a problem. And then just to tie this into active and passive, or not active and passive. Active and passive is, as far as antibodies is concerned, is when you make them or when you don't make them. But what we were talking about with that also was naturally acquired and artificially induced immunity. So the naturally acquired immunity is if the first time you're exposed to chickenpox, you get sick, and then subsequent times you're immune to it. Artificially induced is when you get a vaccine, and that induces the primary response, which ends with making memories, B cells or T cells. And then subsequent exposure to the actual pathogen, you go right into the secondary response, and you never have to suffer from the symptomology of whatever that was because you've been vaccinated to it. That's artificially induced active immunity, okay? As opposed to passive, which I talked about. Passive is when you're getting it from somewhere else, which is getting immunity from your mother through the placenta, or if it's being given to you therapeutically like a snake bite kit. I might not have shown you this picture, but this is just a B cell being activated and a helper T cell helping it be activated, which is why it's called a helper T cell. I said that when I was talking about activation last time, but I might not have shown you this picture. Okay. Now, the rest of what the book goes through kind of goes into some specific topics or special topics, which I don't want to say are not important. I just want to leave that to you to look at. I'm going to concentrate more on the assignments you have on what we've covered in class, the sort of introduction to immunity, the day that we listened to the podcast, and then the innate response stuff that we talked about, and then with the adaptive response stuff, concentrating on the life cycle or the maturation of T cells with positive selection and negative selection in the thymus, and then the activation of lymphocytes, whether they're T cells or B cells, leading to the production of memory cells and active cells that go out and do their job that I talked about and sort of wrapped up today. The other things here are important to look at. The part 1 and 2 assignments are usually made from questions over the entire chapter, and then in part 3 and part 4 where I'm sort of concentrating on certain things, it'll be more on the stuff that I've covered in class. Do understand that there will be questions about the podcast. So I used, 30 minutes worth of class time to go through that because I think it's a useful sort of introduction to thinking about the immune system in different ways than just the strict textbook approach. And to introduce you to some alternative ways of thinking about immunology, like the hygiene hypothesis idea. You'll have questions in part 3 and 4 that go back to that. If you'd like to review it, you have a link to the podcast. You can listen to it again. What I wrote on the screen is in a YouTube video that captured. The video that I captured for this actual class didn't work. It didn't get any of the video stuff. But one of my other classes, I did that. And so if you go to the YouTube channel, you'll see a little thumbnail for an immunity lecture. And I think the thumbnail actually has the hookworm, the teeth in the mouth kind of thing. but you should be able to find it there if you want to see what I captured from the screen that I was writing during the podcast. It is available for you to see again through the YouTube channel. So, okay. So that wraps up the immune system stuff, which wraps up the unit that's on fluids and transport. And so the next thing we're getting into is this unit that's energy, maintenance, and environmental exchange. This unit is basically covering the respiratory, digestive, and urinary systems. And they're interrelated because they all handle some very similar processes. So I do want to kind of talk a little bit to begin with what ties them together to some degree. So we're now in the respiratory system week folder, and then we're going to do digestive and then urinary. In the urinary system folder, there's going to be one of those overview assignments. And the overview is going to be about these three systems together, just how they're similar and how they handle their various problems to kind of work together so the body can exchange materials with the environment around us, how we take in nutrients, how we get rid of wastes, that kind of thing. So that's going to be tied up with an overview assignment once you finish the urinary system assignments, sort of like we had the cardiovascular one and the communication regulation one earlier on. The same kind of assignment there will come after the urinary system. So we're doing the respiratory system in lab, and we're doing it over 2 labs. The first one was about the anatomy of things, and the second one is about how breathing works to some degree. So in the interest of time, I'm not going to repeat that stuff so much here. I just want to draw your attention to the book a little bit. I do sort of mention this in lab, but I want to point it out again. In the book, when they're talking about the respiratory tract or the respiratory system in general, it talks about the conducting zone, which is really the respiratory tract, how air is conducted in and out of your body. And then the respiratory zone, which is really at the end of the respiratory tract when the air gets down deep inside the lungs, the alveoli. This is where we're talking about the exchange of oxygen from the air to the blood and carbon dioxide from the blood to the air. That's what's happening in the respiratory zone. Okay. So when I'm talking about the anatomy in lab, I'm concentrating more on the respiratory tract and the upper and lower respiratory tracts, which is essentially the same thing as a conductive zone in what the book is covering, although I end that discussion of the respiratory tract with the alveoli, which the book is covering the respiratory zone part. In the lung section, it talks a little about the structure of lungs and the pleura, which is the serous membrane associated with the lungs. And I talk about that in the second lab where we're talking about breathing. So I did that for the, have I done that for both groups? Yesterday in lab, today's Thursday, yeah. Yesterday in lab, the Wednesday group, we did the breathing thing, right? Monday group, did we do the breathing thing?
We did use the spirometer.
Yeah, okay. For some reason, I was thinking that one of you was doing it next week, but no, we're good.
Didn't do that.
Didn't do the spirometer?
We didn't do the breathing exercise.
Okay, I'm right, yeah. Okay, so Monday group, you did the spirometers. Wednesday group, we just looked at the models and stuff like that. Okay, so the Wednesday group, I haven't done the breathing stuff. And so that's what we're going to be doing next week. I knew I hadn't gotten to both of them. That snow days have just completely thrown me off. Okay. So I'll be talking about the pleura, the membrane that surrounds the outside of the lungs in lab on Wednesday, because that fits in with the breathing a little bit. And then there's specifically a section that's about the process of breathing. To understand that a little bit, we need to talk about pressure, and so I'll cover that in lab too. The control centers here, I want to cover that in lecture, but I don't want to do that now because I want everybody to have had the lab on how breathing works. So we'll deal with that later on. Yeah. So what I want to concentrate on in lecture is about the gas exchange stuff, which I'm not going to get to right now because I first want to give you some sort of general overview stuff about the respiratory system. Oh, and I also need to show you a picture, which actually I can do the two together. So I'm going to go to my presentations and in the respiratory, system presentation here. Now, I remember when I was talking about this yesterday with the Wednesday group, I specifically mentioned I'm going to show you this other picture in lecture. I actually said to the Monday group when we were doing the anatomy that I was going to show you this. But this is a plastinated picture of the bronchial tree. Okay? So plastic's been inserted into or injected into the trachea up here. Actually, for the sake of recording, let me go to the mouse. Okay, so plastic's been injected into the trachea here, which is translucent. We can kind of see through that. We can see the black background behind it. And that extends down into the bronchi, where it first branches off of the trachea. These are what we call the primary or main bronchi. One goes to the right lung, one goes to the left lung. And then in the lung, there's another branch to the secondary or lobar bronchi, which in this is also contained in this sort of translucent plastic. The lobar bronchi go to the various lobes of the lungs. Okay. How many lobes are in the right lung? 3. How many lobes are in the left lung? 2. Yeah. Just the left lung being on the same side with the heart is a little smaller, so it has a little less space in it. So there are 3 branches on the right side and 2 branches on the left side going to the various lungs. And then within the lungs, within the lobes, there's another layer of branching or another level of branching called the tertiary bronchi. Tertiary means third level. They're also referred to as the segmental bronchi. And that's what's plastinated with colored plastic here. So we can see all of these green bronchi down here. We can follow them back up here to where they branch. Or is this kind of a light blue color maybe? Yellow, orange, red, light blue, medium blue, white, brown maybe? I don't know. Those are different segments of the lungs. Air traveling to that part of the lung is only traveling through that tertiary bronchi and then all its subsequent branches off of that. If something blocks this green bronchus right here, then none of this tissue is going to be functional. None of it's going to get any air. But all of the rest of this lung, and of course the other lung, are fine. They're not compromised by something blocking that one segment. And I really like this picture because it really kind of highlights that the pattern there. The model that I had in lab is meant to show you the same sort of thing. It's not a very extensive bronchial tree. It just shows sort of that tertiary level of branching coming off. And those are old models, so some of them are starting to break off. You can't see all the different colors. But this is really good because you can really see the shape of the entire lung here. And so these are all of the bronchi going out to all the different nooks and crannies and corners in the lungs. And we can see that they are really segregated by color there. So, great. Okay. The other thing I want to talk about is sort of the functions we associate with the respiratory system. The first thing, pulmonary ventilation, means breathing, okay, to ventilate, to move air in and out of the lungs. And then external respiration means gas exchange. It's external because it's the gas exchange between the blood and the air in your lungs. Okay. So that's oxygen into the blood, carbon dioxide out of the blood. Transport of gases means moving those gases from the lungs out to the tissues, or from the tissues to the lungs. Okay. So that's what's happening in the circulatory system. Internal respiration is, again, gas exchange, but here, internal is referring to what's happening between the blood and the tissues. So oxygen in the blood goes into the tissues, carbon dioxide from the tissues goes into the blood. And then finally, I mentioned cellular respiration here because this is really the point of the respiratory system. When you hear respiratory system and respiration, you probably think about breathing, but that's not really what it's about. It's about gas exchange. It's about getting oxygen and carbon dioxide moving in and out of our bodies, respectively. And cellular respiration is just about how those gases are used at the cellular level. And it really refers to the chemical reactions that we process energy through. So let me go back here. And what we're talking about is C6H12O6. I can't put subscripts on here, but this is a chemical formula. Does anybody know what that's a chemical formula for? C6H12O6? Yeah. Glucose, sugar, both appropriate answers. We usually say glucose, but actually it's the same chemical formula for the two other main monosaccharides, which are fructose and galactose. Okay. So it could be any of those, but generally we usually think of it as glucose. Okay. And if we add to that 6 O2s, oxygen gas, we can yield 6 CO2, carbon dioxide gas, and 6 H2O, water. Okay. If you look at all the atoms represented here, the carbons, the hydrogens, the oxygens, there's an equal number on both sides. This equation, what we call cellular respiration, is how glucose is completely broken down into carbon dioxide and water. Okay. This is done to generate energy, or to, I should say, harvest energy from the glucose. That energy is used to make ATP. Okay. ATP is not in this particular reaction because it's a separate reaction. ADP, adenosine diphosphate, plus inorganic phosphate. Again, I can't put a subscript in, but P subscript I is inorganic phosphate. whoops, yields ATP. Okay. It requires energy to put a third phosphate onto ADP. Okay. And if we reverse that reaction, if we take that third phosphate off, that energy gets released. The energy necessary to make the ATP in the first place is energy that's released by breaking down the glucose in the presence of oxygen. Now, in the list I just showed you there, cellular respiration was sort of at the bottom of the list of functions associated with the respiratory system. This is not happening in the respiratory system itself. This is happening all over the body, but it's made possible because of the respiratory system to some degree. Okay. Actually, this is a good example of where these three different systems come together: respiratory, digestive, and urinary. Okay. The respiratory system is about getting the oxygen and the carbon dioxide in and out of your body. And that's what we're going to concentrate on today and Tuesday. The digestive system is where we get glucose or other nutrient molecules. But like I said, we usually talk about glucose primarily here. And then water as a waste product here, we're going to get rid of in our urinary system. Okay. Now, the urinary system certainly does a lot more than that, but that's sort of rounds out talking about what's happening with this reaction here. Okay. Now, this reaction is actually not happening by itself in your body. The amount of energy released here is really pretty significant. And if this were to happen in our body, if we just took glucose and added oxygen to the mix and got the reaction started, and it gave off CO2 and water vapor, or water as a waste product, a lot of energy would be released. Okay. This reaction, actually, when it does happen, that's fire. Okay. Wood, when it's no longer part of a living tree and it's been dried out appropriately, is pretty much all made out of glucose. Now, plants make glucose, plants or trees, whatever you want to say, make glucose in photosynthesis. They make a lot of glucose. And so they use glucose as a structural element in their cells or for their cells. And the main structural element that we see glucose in this form is the cell wall. Cell walls are made out of this rather complex polymer of glucose called cellulose. If you have wood and it's dead, so the cells are no longer there, all that's going to be left over are the cell walls, which is made out of cellulose, which is made out of glucose. So if you light a piece of wood on fire, you're burning the glucose, essentially. Now, there needs to be oxygen present for the fire to stay burning. If you remove the oxygen source, you put out the fire. And that's largely the whole point of putting out fires. You either douse it with water so no oxygen is available, or you cover it with something like, if you wrap somebody in a blanket when they're on fire, you put out the fire. A fire extinguisher is just throwing some sort of chemical that will block the oxygen getting in there so it doesn't burn. Okay. But if you are burning wood, if it's a fire in your fireplace or something like that, as the wood burns, it ideally is releasing CO2 and water vapor. Now, not just water because of the temperature, it'll necessarily be in its vapor form, but H2O nonetheless. And that's essentially what smoke is. Now, smoke can contain other things because sometimes the wood that you're burning is not just pure cellulose. It probably isn't. It's probably going to have other things, some lipids and that sort of thing that might be sap that's left in the tree or whatever. And as those burn, they're going to give off different chemicals. And that's going to make the smoke thicker and darker and that sort of thing. But if we just had pure cellulose, pure glucose, and we lit that on fire, what it would give off would just be CO2 gas, and water vapor. Okay? And it would give off a lot of energy in the forms of heat and light. You don't want that happening in your body. Okay? So this reaction never actually takes place in your body just by itself. We control it by distributing this process across a couple or few dozen different chemical reactions. We're going to talk about those chemical reactions in more detail when we get to the digestive system. I'm not going to deal with it right now. Here we're just thinking about getting the gases in and getting oxygen, getting carbon dioxide out. Okay. So in my presentation here, I do some of the anatomy. I want to go over to this section here and talk a little bit about gases. Now, the first thing about gas is this pressure-volume relationship, which is what I talk about in lab. So Monday people, I talked about this. Wednesday people, I'll get to it next week. What I want to talk about instead are a couple of laws about gases. One's called Dalton's law. And to explain this, I want to play this little video. Now, this is just a YouTube video that I've embedded here. And it explains, whoops, sorry. I don't know why it's cutting off the edge of this a little bit. It should have some space in it. But this guy's going to explain what Dalton's law is. And as you might guess from the colored balloons behind him, as far as production value is concerned, it sort of looks like it's meant to be for kids. And it has some sound effects that are kind of silly too. And you can tell from his face, he thinks they're kind of silly too. But what he's talking about is not something that you'd have, grade school kids trying to learn. Dalton's law is a little bit more complex and does require, an understanding of algebra concepts or something like that to see what it's saying. So it's a kind of silly video, but what it's saying is important. So let me. Why is it not playing? There we go. Would help to have some sound.
What I have here is a balloon filled with ordinary air. Air is a mixture of gases, and you probably have thought that air was mostly oxygen. After all, oxygen is the piece that you're most interested in. You need it to survive. In fact, air is mostly nitrogen, N2. It's about 78% nitrogen and only 21% oxygen. What we're going to be talking about in this section are mixtures of gases. And the question that we're going to consider here is, we have a balloon. There's pressure from the gas on the inside of the balloon exerted against the outside of the balloon. If it's a mixture of nitrogen and oxygen, how are the forces of the individual gases related to pressure, and how are they related to the total pressure of the balloon? So in other words, each of the individual gases presumably exert some pressure, and how is the component of each of the individual gases related to the total pressure of the balloon? I'll get rid of this. To answer that question, what we need to do is define a quantity first called the partial pressure. Okay? So here we have a mixture of gases. It's inside a container of volume V, and each of the individual gases is indicated by different dots. So we've got some red dots and we've got some blue dots. What I want you to think about is, suppose we took everything out of the box except for the red dots. So we have a quantity of gas A in the volume V, and we can measure with this pressure gauge the pressure due to A just in the box all by itself. And we're going to call that quantity the partial pressure of A. So again, piece of A, the partial pressure of A, is equal to the pressure due to A as though it were alone inside the container. Okay? This is in the context, again, of a mixture of gases. Well, we can do the same for B. B, let's say, are the blue dots. We can have just B inside the box. We can define P sub B as the pressure due to B alone in the container. And now we get to Dalton's law. John Dalton, in the early 1800s, discovered that the total pressure exerted by a mixture of gases equals the sum of the pressure of the component gases. And this is now known as Dalton's law of partial pressure. If we look at an algebraic representation of Dalton's law, it says that the total pressure inside the container is equal to the partial pressure of A plus the partial pressure of B plus the partial pressure of C plus whatever else might happen to be in the container. And the really important point of this is that basically what this says is that A is oblivious to the presence of B and C. And similarly, B doesn't know that A and C are in there. So the gases don't even interact at all. And that is a tenet of the ideal gas idea, that the particles are so far apart that they're not really even interacting with each other. And so it really doesn't much matter whether the gas.
Okay, so this is just an excerpt from a larger lesson, but it covers what I wanted to explain here. So Dalton's law Okay, so Dalton's law is basically that the total pressure of a mixture of gases is equal to the individual partial pressure of each. So that's a restatement of Dalton's law there. And he represented that by the pressure of the total mixture is equal to the pressure of gas 1 plus the pressure of gas 2 plus the pressure of gas 3 plus et cetera, et cetera. Okay. That's the idea of what Dalton's law is. Okay. Now, if we apply this to the air in this room right now, I am going to make a couple of assumptions. One, if we were at sea level, okay, and that's important because the pressure of air changes due to elevation. Okay. So let's pretend that we're at sea level. So the pressure of the gas in this room, the air in this room, is 760 millimeters of mercury. Okay, that's air pressure at sea level. Oh, just as an aside, does anybody own a Samsung phone? Do you have a barometric sensor on your phone? This is a cool new kind of sensor that I've heard about, and I think Samsung phones are the only ones that have them. I'll have to ask somebody at some point. I was hoping somebody, oh yeah, I've got the app that does that. Oh, cool. If there is a barometric sensor in the phone, what we could do, what I'd love to be able to do, is to measure the pressure of the air here on the 5th floor versus the pressure of the air down on the ground level here. And hopefully the app would also be able to compare that to sea level, which we're probably fairly close to, but I don't know actually what the elevation is in Springfield. And we are actually at the highest point in Springfield. The Armory was built on the highest hill in the city. And so like the flag that's on top of the Armory right there is supposed to be sort of the highest thing in the city. Whether it is or not, I'm not sure, but at one point it was. So anyways, this is air pressure at sea level, so we'll work with that. And it is really the summation of all the partial pressures of everything else. So the partial pressure of nitrogen plus the partial pressure of oxygen plus, whoops, I'm not doing plus signs in there, plus the partial pressure of CO2 plus the partial pressure of H2, whoops, 2O, I keep doing that, plus the partial pressures of other things in this air around us. Those four are the major components of the air that we breathe. Now, when we're representing partial pressure, we actually would be using subscripts. So the particular gas, the abbreviation used for the gas would be a subscript to P. So it'd be partial pressure of nitrogen plus the partial pressure of oxygen plus et cetera, et cetera. So that's really how we depict it. I just can't do subscripts in the field that I'm typing in here like that. Okay. Now, the way that we figure out a partial pressure, because we can't actually remove everything from the air around us except for the nitrogen. Instead, what we can do is we can take a sample of the air, not us, we're not going to do that, we know the values, and measure how much of it is nitrogen and how much of it is oxygen, et cetera, et cetera. And then the percentage of the mixture that's one thing can be used to figure out what percentage of the overall pressure is that for that gas. For example, nitrogen makes up, like you said, about 78% of what's in the air we breathe. And so the partial pressure of nitrogen is just 760 millimeters of mercury times 0.78, okay? And that's going to give us the partial pressure of nitrogen. If I remember correctly, that comes out to 593 millimeters of mercury. Might be off by a little bit, but it's somewhere just a little bit south of 600. Okay. And we can just figure that out by calculating the percent contribution to the overall thing, rather than having to actually measure it. We did the same thing with oxygen. Oxygen is 21% of the air that we breathe, and its partial pressure is 10. Wait, that's not right. Yeah, 159. That's right. It's actually 20.9%, so that comes out to 159, not 160. But and so we can do the same thing for carbon dioxide and water vapor too. Carbon dioxide is a very, very small amount of the air that we breathe. It's a fraction of a percent. So, and I think it's 0.03 or something like that. So it ends up being a very, very small partial pressure, but it is there. And then water vapor is like 0.5% or something like that. So it's a little bit more than carbon dioxide, but it's still very small. So we know what the partial pressures are of the various gases in the air that we breathe because we already know what the composition is and we know what the total pressure is. Okay, so those are givens. If the composition changes, then the partial pressures are going to change as well. And changes are going to happen because of what happens in the next thing here. Which is, I want to talk about Henry's law. Now, Henry's law is really talking about how gases interact with solutions. When we're thinking about things at the molecular level, O2 could dissolves into a water solution. CO2 dissolves into a water solution or something like that. The thing is that if we're going to mix a gas into a liquid, we can't just measure out some stuff on a scale and pour that into a water solution and mix it up like we would to make, a salt solution or something like that. Instead, it has to do with how the gas diffuses through air and water. And essentially it diffuses equally across the two. Henry's law just says that, and at a given temperature is kind of not terribly important to us because we're always kind of thinking about body temperature, 37 degrees. But at a given temperature, the amount of gas in solution within a liquid solution is proportional to the partial pressure of that gas. Okay. We don't talk about the concentration of oxygen, the concentration of carbon dioxide when it's in solution. We just talk about its partial pressure. So if we had a beaker of water here sitting open in this room, the partial pressure of oxygen, like I just showed you, is 159 millimeters of mercury. So in that open beaker of water, the partial pressure of oxygen in that solution would also be 159. Okay. Oxygen molecules are moving around randomly in this room, and they would diffuse into the water, and they would be equal in distribution with this between the air and the water. Now, this picture kind of suggests this. This is out of a different textbook, and if I were to draw this, I would have had 3 different pictures just to really show you what's going on. This is a container with a lid, okay, a beaker with a plunger on it or something like that. The dotted line represents the location of the lid to begin with. And when it was in that position, the space below the lid was equal in volume to the space that's taken up by the liquid down here. And the partial pressure of the gas represented in here was equal to the partial pressure of gas within the solution. Okay, they're at equilibrium. So what happens is if we push the top down and make the volume smaller, the pressure in the air component here is going to go up. Okay, and so more gas will diffuse into the liquid until it's at equilibrium, so that the pressure in the air and the partial pressure in the water are equal. Okay, and this is sort of like what you see in a unopened soda can. When you're bottling soda, when you're putting it in the can, you force carbon dioxide into the can with a hose, and it'll be under high pressure, and that will then work its way into the soda solution. And then you cover the can, so you close up the top of the can, and the pressure of carbon dioxide inside the can, whether it's in the air or the water phase, is much higher than what's outside the can, but it's consistently the same pressure all in the can. If you then open the container, so if we take the lid off of this beaker, suddenly the pressure in the air component drops drastically because it's now continuous with the outside, and the gas that's dissolved in the liquid phase will come out until it reaches equilibrium with the air that's now open to the outside. And so in an open soda can, the carbon dioxide that was forced in earlier will all come out. Okay. What we're looking at here is flat soda. Okay. The process between the unopened can and the open can at equilibrium, that's when the soda is bubbling, because all of that carbon dioxide in the water rushes out as there's a huge change in the pressure in the air phase, and it comes out in bubbles. Okay. So when you think of soda, you think of the carbonation in soda. That's really, once you've popped open the soda can, that's all of the air rushing out is the carbonation. Once it reaches equilibrium with the surrounding air, then the soda is considered flat because it has no more carbonation or carbon dioxide in it than any drink that you might have. Okay, plain old water is going to have a little bit of carbon dioxide dissolved in it because there's carbon dioxide in the air around us. But during the bottling process or canning process, extra is forced in there so that when you do pop it open, that carbonation is what makes it all bubbly, okay, which apparently is a wonderful thing and people love to have soda. So that's Henry's law is describing that. And it's important here, not because we're worried about soda. I'm just using the soda can, one, because it's in the picture, and two, because it's probably something you have some experience with. But really what we're talking about is air moving from gases moving from air into the plasma of our blood. Okay, that's where Henry's law really applies here. Okay. And so in the book, we're going to get into next the gas exchange portion. And it starts off, and I've already kind of done this, talking about gas laws. Here's the table of partial pressures of atmospheric gas. So there's nitrogen, oxygen, water vapor, carbon dioxide, and other things that are out there. Nitrogen's 78.6%. Multiply that by 760, you get 597. So I was off a little bit in what I wrote, but I was doing it from memory. Oxygen is 20.9%. Multiply that out, you get 158.8, which rounds off to 159. And so you can see all of these here. And this is sort of the book's representation of the same thing that was in the video. If we just had oxygen or just had nitrogen by itself, we get its partial pressure. If we put them together, we get a total pressure there. And the numbers they're using here are the numbers up here. And so this mixture of oxygen and nitrogen doesn't have any of the other components in gas. So that would be the pressure of just those two by themselves. Okay. And then the solubility of liquids and gases in liquids is about the Henry's law stuff I just said there. The ventilation stuff we'll talk about more in lab. And we want to get into the gas exchange stuff, which is external respiration and internal respiration. And obviously, we're not going to start that now because we're to the end of today's class. So next time, we'll pick up with external respiration and cover internal respiration too, and also how we regulate the respiratory system, which will go back to the breathing part. So that's it for today.
Before we all leave, I have a quick question.